and Dr. S.K. Bandopadhyay to kindly preside as chairs. You have already been acquainted with Dr. S.K. Bandopadhyay, who is a distinguished pharmacologist with over 40 years of teaching and research experience. To jointly steer this prestigious session, we have Dr. Shiv Prakash, a highly distinguished leader in pharmacology and drug discovery, currently serving as president of IPS, chief advisor. A prominent academic and industry figure, Dr. Prakash is a former chief editor of Indian Journal of Pharmacology and past vice president of IPS. He has authored over 100 research papers and a book holds multiple patents and is a recipient of prestigious awards including Udyog Ratna and Entrepreneur Excellence Award. This symposia features 3 distinguished speakers presenting clinical and molecular breakthroughs. I would request honorable chairpersons to introduce our eminent speakers for the session.
Okay, good afternoon, everybody. Our first speaker of this symposium is Dr. Anil Gulati. Dr. Anil Gulati, Dr. Anil Gulati is an eminent pharmacologist, innovator, and entrepreneur, recognized for his pioneer contribution to translation drug development. He is the founder, chairman, and CEO of Pharmas Corporation and holds eminent emeritus position at Midwestern University and University of Illinois in Chicago. A graduate of King's George Medical College, MBBS as well as MD, he earned his PhD in pharmacology and therapeutics from Erasmus University, Rotterdam, and is certified by the American Board of Clinical Pharmacology. Professor Gulati's groundbreaking research led to the development of Centhaquine, a novel resuscitative agent for hypovolemic shock, and sovatildide and endothelin beta receptor agonist for treating cerebral stroke. His innovative approach of stimulating dormant regenerative cells to repair damaged brain tissue has opened new frontiers in neurotherapeutics, receipt of several prestigious awards including the International Ranbaxy Research Award, Paul R. Dawson Biotechnology Award, and Little John Award. Professor Gulati has over 300 publications, 60 patents, and has mentored countless students and physicians worldwide, exemplifying excellence in scientific innovation and leadership. With this brief introduction, I request Professor Gulati to deliver his lecture.
Hello, can you? Yes. Thank you, chairpersons, and it was almost 8-9 months ago when Professor Anil Kumar approached that you should be coming here. So, and Chandigarh happens to be my in-laws place, so I said, yes, I'm coming 100%. So here I am, and it's always a pleasure to meet everyone. Although my trip is short, but it is going to be sweet because meeting everyone is old memories which come into picture. So I have been professor almost throughout my life, and then I became emeritus. During my inventions, the inventions are disclosed to the university, and then I started a company to develop those as a drug. So this company Pharmas was founded in 2010 by, I'm the founder, and then now it has grown to the extent that we have several products which are in market, at 2 of them in market now in India, and phase 3 in the US. One is for acute cerebral ischemic stroke, and we have partnered with Sun Pharma. They are our sales and distribution entity. Other hypoxic ischemic encephalopathy, phase 2 is going on in India. Alzheimer's disease, phase 2, 80 patients completed in India. And the product which I am going to talk about is Centhaquine, which originally is from CDRI, where I worked initially after my graduation. Hypovolemic shock, it has launched in India, and phase 3 in the US. ARDS, acute respiratory distress syndrome, we have a phase 2 IND approved by the US FDA. Septic shock is another indication which we are pursuing. And so I'll focus more on Centhaquine today. And the uniqueness of this is that this product is free of arterial constriction. It is marketed under the brand name of Lifaquine. So it's a new paradigm in the treatment of shock because it is free of arterial constriction. I just want to emphasize again and again. So how does it increase the blood pressure? This will be a very interesting story as we go along, that in spite of no arterial constriction, rather it has little bit of arterial dilatation, but still it can increase the blood pressure in a patient with shock. So patients in shock where there is excessive blood loss or septic shock, there is very high mortality, somewhere between 15 to 20%. Even in some of the underdeveloped or partially developed countries, this mortality can go up to almost 35%. So what happens is decrease in cardiac output, hypoperfusion, multiple organ failure, and death. That is the critical. So basically blood pressure goes down, oxygen level goes down, and organ failure, and ultimately death it takes place. So the current treatment is patients are given fluids, crystalloid, colloids, and blood products and all that, and fluid, if there's not enough, vasopressors are added, which are arterial constrictors. Some norepinephrine, we all know, is for ages it has been used. Then there is vasopressin, angiotensin was recently launched. So all of them are arterial constrictors. So arterial constrictors, they actually reduce the tissue blood perfusion. There is a lot of beta activity and other kinds of cardiac arrhythmias. They are another problem, so they have to be titrated very carefully. Fluid extravasation and vasopressor infusion requires careful titration, and in US most of the time these have to be given through a central line. So that means a small procedure has to be done, and so then the central line is put in the big vein, and then the infusion is done. So this is the first experiment which we did for Centhaquine, which was very informative. So I always use this slide. So here is the blood pressure, then the massive blood loss is produced. This is the Vickers model where there is blood loss, so the blood pressure goes down. This is the mean pressure. Heart rate, you can see that once blood loss is there, it goes down. When we give Centhaquine, look, the blood pressure is going up and heart rate is going down. So this observation was very alarming to us, that blood pressure is going up and heart rate is going down, and most of the arterial constrictors or vasopressors used today, their heart rate also goes up, not down. So that was, so we did pressure volume studies, and Dr. Manish Lavale is here. He is now the Managing Director of Pharmas India. So he did these experiments way back many years ago, where this width gives us the stroke volume, and you will see that in the next slide, this is the sham pressure volume loops, and here there is blood loss. This is vehicle, this is Centhaquine, and you can see that the stroke volume significantly improves. Here it's going back and ultimately death. There was no animal survived till 240 minutes. So these studies were done in Greece independently by Professor Theodore Xanthos, and what he found was in a swine model 50% of blood volume was taken out, and we can see that the fluid administered in vehicle was close to 2.2 liters, while in Centhaquine it was only 300-400 ml. So without much of fluid infusion, the resuscitation could take place, and 2 hours later in vehicle only 2 animals survived, while all of them survived 2 hours, and even 24 hours 9 out of 10 animals survived. So these were very positive results which led to our further studies now to see the concept. So you can see that the cardiac output, when there is blood loss, it goes down, and in Centhaquine cardiac output is significantly improved. While what happens to vascular resistance, it is decreasing. So cardiac output is increasing, vascular resistance is decreasing. When vascular resistance goes down, the blood pressure should fall, but the blood pressure is going up. That means the major contribution is coming from cardiac output. So lung wet dry rate, these findings were also positive, that 4.8 and 3.08. Horowitz index also significantly improved. Mortality also, you can see that swine, it was quite a big difference between the 2 groups. So all of these were pointing us to something which we thought that let us explore further, particularly cerebral blood flow, and you can see when blood loss is taking place, there is decrease in perfusion, and with Centhaquine, the perfusion is increasing as we go along. So now you can see that quite a significant increase in cerebral blood flow. Now this is important because brain has very intense autoregulation. In spite of that, the blood flow is going up in the brain. So free of arterial constriction, cardiac output increase is the dominant effect which leads to an increase in blood pressure, and vascular resistance is decreased. So the new concept of resuscitation was put in. Now the idea was that there is 30% of the blood is on the arterial side, 70% is on the venous side. So venous blood pools very rapidly, and when a patient goes in shock, this pooling further increases. So already 60-70% of the blood is on the venous side. When shock occurs, it further goes up. Centhaquine, what it does is it moves that blood towards the arterial side. So veins which are compliant can accommodate, and large amount of blood is on the venous side which can be moved towards the arterial side. So Centhaquine, where it acts, so we did a lot of studies, and various receptors were identified. Ultimately alpha-2b, and 95% of alpha-2b receptors are on the venous side. So it is constrictor of these, and then there are alpha-2a receptors in the central nervous system which are decreasing the sympathetic drive. Therefore there is decrease in vascular resistance. But the key thing is increased venous return to the heart. That will lead to increased cardiac output, and heart pumps more blood per beat. That means stroke volume is increasing, and predominant effect is cardiac output increase, which is leading to increased blood perfusion. So this is a human study in which cardiac output and inferior vena cava diameter was measured. So you can see that there is very good correlation between cardiac output in humans with hypovolemic shock and inferior vena cava diameter. So these findings have been published now, and you can see that inferior vena cava diameter 0 minutes, 60 minutes, 120, 300 minutes in humans, it is gradually increasing. So what we did was we tried to correlate or regression analysis between stroke volume and inferior vena cava, and you can see a very, very good correlation between stroke volume. So in humans also, besides animal models, in humans also we are proving that it is the stroke volume which is contributing towards an increase in blood pressure. So here with blood pressure also, inferior vena cava diameter, it is correlating very well. So there is venoconstriction by Centhaquine, and that venoconstriction leads to an increase in cardiac output, and in alpha-2a activity in the brain leads to a decrease in vascular resistance. So we did phase 1, phase 2, phase 3 in India, and ultimately the drug has been launched. So these are the phase 2 results which I'll show you, and you can see that the demographics, they were similar between the two groups. I won't go in those details. The amount of vasopressors infused in the first 48 hours was from 9 to 3. So there is quite a decrease in the use of vasopressors, and we know that if we use less of arterial constrictors, the patient's adverse events are much less, and survival also is way better. Time in ICU also goes down. It is not significant, but there is a trend that it is going down. Hospital stay, ICU, then time spent on ventilator from 1.9 to 0.89. That also, the trend is that it is going down, but because of error, it is not significant. Blood pressure, you can see that in a patient, the green is Centhaquine and red is standard of care, and we can see that the blood pressure in Centhaquine group is much better than in, so systolic blood pressure and similarly diastolic pressure, both are improved. Base deficit, which is an important indicator for mortality, we can see that in Centhaquine group, it becomes base positive instead of deficit. Lactate levels, very important, which are very key indicator of mortality in a patient with shock. We can see that huge decrease in lactate level. I think Dr. Manish was one of the, when he was doing those experiments, he constantly noticed that lactate level goes down, and that led to the discovery of infiling patterns and ultimately launch of this drug. So in individual patients, we see the data that lactate levels in a control group p-value is 0.29, while in Centhaquine group it is 0.028. Now there were, I think there were two patients which were very well matched between the two groups, and this is patient 10 and patient 12. Patient 10 in the control group and patient 12 in Centhaquine group, and both patients were ectopic pregnancy which went bad, and massive blood loss took place. So the age is 24-year-old female, 31-year-old female. This is in the Centhaquine group and this is in the control group. You can see that blood loss, hemoglobin level went down to 9.3, and here it is 3.6. And blood lactate level from 11, and here it is 17, which is very, very severe. And this patient could not survive, while the Centhaquine patient did survive, and almost 40 milligrams of vasopressors were also given. So we had 2 deaths in phase 2 study in the control group, while 0 in the Centhaquine group. These findings were published in this journal, and then we embarked on phase 3. Phase 3 we did, there were 2 groups, and block randomization was used. Because the results in phase 2 were so promising, it was decided that it will be 2:1 ratio, that more patients will receive Centhaquine than the control. So basically 2:1 ratio was followed. So these are the hospitals where the trials were done, and you can see the basic baseline hemoglobin. This is Centhaquine green one. It is equal but little bit less. Hematocrit also a little bit less, and blood pressure in phase 3 also, it is the same trend which we were finding in phase 2, that the blood pressure, systolic as well as diastolic, both are improved compared to the control with standard of care. Shock index here which we measured, this was also quite promising in the sense that with Centhaquine, the shock index statistically, you can see that within matter of half an hour, it started reaching the level of significance, and then it is significant all the way down. So very significant improvement in shock index once again, and shock index in the early 6 hours is one of the most important index for survival. So lactate levels, as we found in phase 2, once again they are decreased. Number of patients with less than 1.5 lactate is also better in Centhaquine compared to control. Base deficit, same finding which we got in phase 2, that number of patients showing base deficit improvement is statistically significant. ARDS, which is an important indicator in shock, also you can see that it reached the level of significance in Centhaquine, while in control it was not. MODS, which is another indicator, Centhaquine, there is MODS, or shock is the patients are recovering much better than in control. So what happened was, in phase 3, all the primary endpoints, 4 primary endpoints, which are blood pressure, systolic, diastolic, blood lactate and base deficit, they reached the level of significance which was statistically less than 0.05. Mortality, 28-day mortality in control was about 12%, while in Centhaquine it was about 3%. It did not reach the level of significance 0.07, but a very big difference in absolute mortality. So we did further analysis of, because phase 2 and phase 3 inclusion exclusion criteria were similar. John Louis Vincent, who is world renowned person in critical care from Belgium, in his editor of 4 or 5 journals, which are all above 30 impact factors. So no pharmacological intervention consistently reduced mortality, and Centhaquine did show that when we did meta-analysis of phase 2, phase 3 combined, that the p-value did reach the level of significance. So there were adverse events. There were total of 9 adverse events in phase 3, 4 there were deaths reported in the control group, 2 deaths reported in Centhaquine group, and there were 2 events with creatinine level increase, which resolved without any issue. There was 1 event of vomiting, so not much of adverse event in a critically ill or dying patients, which was observed. These findings have been published in the journal Drugs, and so to summarize, this is the concept, which is Centhaquine, when it is administered, it has multiple 2 predominant effects. One is that venous pressure goes up, venous return goes up, ventricular filling, and Frank-Starling's law, more blood comes to the ventricle, more is pumped out of the ventricle. So ventricular contraction improves, stroke volume goes up, cardiac output goes up. Heart rate decrease, which is because of CNS effect, also will lead to increased diastolic time, which will lead to increased filling time, ultimately contributing to ventricular filling. Simultaneously, fluids are given in a shock patients, that also will increase blood volume and ventricular filling. So overall, you can see the schematic is that it favors an increase in cardiac output. However, since Frank-Starling's law is involved, we have to be cautious that patients with heart failure, the efficacy may be much less. So in other words, the schematic is alpha 2A activity in the brain leads to arterial dilatation, which decreases the afterload, while alpha 2B activity, venous return, and that leads to increase in cardiac output. And because the afterload is decreased, preload is increased, all that contributes towards improved tissue perfusion. So it is easy, it comes, we just 10 ml vial, we put according to body weight, the volume, and ultimately infused over 1 hour in 100 ml of saline. Now what happened, we took all this data and went to US FDA. So we submitted this phase 3 IND for US FDA, and we were, that happened first time that not a single patient was done in United States. And US FDA approved our phase 3 trial. And this is the design of the study which US FDA has approved. It will be a randomized, double-blind, and primary endpoint is all-cause mortality at 28 days. Secondary endpoints and exploratory endpoints are mortality 60 days, ventilator-free days, more of patient-oriented endpoints which are FDA looks for. So all those are in place now. We have formed a committee to look into the trial. So the difference between the US FDA approved study and the India study which we did, the primary endpoint in India was blood pressure, lactate levels and base deficit. Here it is mortality, and those blood pressure and all that will be secondary endpoints. Inclusion criteria is exactly similar. When a patient comes in an emergency with massive blood loss or in a shock, then the blood pressure, if it is systolic blood pressure goes below 90 and lactate level is above 2, the protocol kicks in for resuscitation. In India, there were patients with massive fluid loss also besides blood loss. In US, the FDA asks us to, etiology should be, if it is not available, then those patients should be excluded. 430 patient size and 7% reduction if we assume, although in India we got closer to 9–10%, then the drug will be approved in the US. Randomization 1:1, futility analysis is there. So treatment, these are all ages old drugs which are used. There is nothing new which has come up. So standard of care is more or less similar between the multiple countries. So ultimately, one major difference is that for Centhaquine, since it is not an arterial constrictor, it can be given through the peripheral vein. While all other arterial constrictors, because of necrosis, it has to be given through the central line. So central line and peripheral, it makes a very big difference because of surgical procedure, and almost an hour is lost putting the central line, and that 1 hour can be very, very critical in a dying patient. So we formed a world-renowned investigator. So Professor Jean-Louis Vincent agreed to be on my committee as an advisor, Daniel DeBacker from Brussels, Matthew Legrand from UCSF, Jan Bakker from Erasmus, Marlene Osterman from London, Ashish Khanna, who comes here, and his father was here in PGI, so he has a lot of attachment to Chandigarh also. So they are our committee, and
now, in the future, I may add this slide. Centhaquine has a very interesting profile as an alpha-2b stimulating agent, and it has a totally different mode of action. But the idea of vasoconstriction, I believe, is an excellent idea that could help to limit fluid administration and perhaps limit blood transfusion, or at least maintain a better hemoglobin level, because it's the patient's blood which is put in the circulation. And there is no vasoconstriction, as we heard, and there could be a small decrease in heart rate. So these are a number of potential advantages. And so in the future, in a few years from now, we may actually put it at the very beginning of our management. We could say, okay, start fluids and give already some.
So takeaway from his message and from the committee is that very different mode of action compared to current vasoactive agents. Venous constriction, very good idea, and it is the patient's own blood which is being increased, which is used to increase the circulating volume. So hemoglobin levels are much better maintained than in infusing fluids. No arterial constriction, decrease in heart rate, and his advocacy is that begin management of shock patient with the start of fluid and Centhaquine. So now where are we right now? So we know that FDA has approved. We formed the committee and we are planning to embark on this study. In the meantime, we also have a phase 2 IND from US FDA approved for ARDS, and it's a 80 patient study. Professor Landoni is the leading person, and this we have already funded the trial, so it will be done in Italy, and he'll be coordinating all the activities. So 15 patient study for cardiac output was done, and that is the data which I showed you, where the very good correlation between inferior vena cava diameter and stroke volume. Phase 4 study, 400 patient study is ongoing in India, out of which 344 patients have been enrolled till date. Then in Turkey, there is another study. This has been completed. We are about to get the results from this study. And then there is a septic shock study which is being started in the Erasmus University in Brussels. So a lot of coverage takes place, and thank you very much. I will be leaving soon, but my emails, university emails and company email, they are all working. So please do not hesitate to contact me because as soon as I'm done, I have to leave back to Delhi and then to Bombay. So thank you very much. Any questions?
Hello. Yeah. Thank you. Thank you. Thank you very much, sir. There are two more excellent speakers in queue. We will allow one question. Anybody, one question. Please.
Yeah, I think that compound is still in early stages of clinical trial, and the concept is a little bit different. There is more of volume preservation rather than increasing the blood pressure through resuscitative measures. So that concept is more of volume preservation, that the substance, when it is given, it leads to preservation of the volume and not extravasation of fluids and all that. So a little bit different from, it can be used complementary down the line.
In one word, I will ask you just one question. Will you consider it as this drug Centhaquine as a cardiac stimulant or a cardiotonic?
Yeah, so I will consider more of a non-stimulant, more of a cardiotonic, but there were initial studies in CDRI wherein higher doses. Yeah, it is more towards venous side. That is the whole concept. So in higher doses, it was shown, Dr. Shrimal, he did some experiments in cat model where he found that in higher doses, it can increase cardiac output through contraction.
Thank you very much, sir.
Thank you.
Now I request our session chairs to graciously present a token of appreciation to Dr. Gulati.